Are we on the same page?
So far.
Because we have a lot of these to work. Are you okay with that?
Are we working them in class?
No, we only have 2-3 hours in class. It takes more than 3 hours to work because you're going to have to work through the problem. Your assignment, what you have to do, you may need to write this down because I'm not sure I wrote it on the exercises. For this particular assignment, you should do this on a computer. If you don't do it on a computer, you should do it on graph paper. So your assignment is going to be that you have to graph the gene frequency of the P and the Q. Because it's going to be, we're going to be looking at gene frequency over a period of time. So you're going to have to graph the change in the frequency of P and graph the change of the frequency of Q over time. So everything in that problem is like sometimes it's 1 year apart, and so you have to set up a graph and set it up that way. Let me tell you now that the x-axis is going to be the time. All right, the x-axis is going to be the time. The y-axis is going to be the other variable. So when you set up your graph, so say many years ago, before computers were so plentiful, you had to do this on graph paper. But now most people will do it using Excel and on a computer or whatever graphing program and do your graph. What will not be tolerated, meaning that if you turn it in and like hand drawn, this person and I can't even see that. It's invisible. I'm making my point. So don't do a hand-drawn graph. It's just a big zero. Okay? You have to graph it on, do this graph on graph paper or on your computer. So don't hand draw it. Like you came in today, if you don't have a printer attached to your computer, just don't turn it in because it will not be accepted. Okay? So you're going to have to do, you have a problem with squirrels and you're going to have a problem with Drosophila. And where you have to graph the change in gene frequency. Let me divert a minute. I finally thought of the other term. I'm sorry, I just couldn't think of it. Genetic drift.
That's the other one we need to look up besides bottleneck.
Yeah. This is the other term that I need you to look up. And you can put that on your assignment that you're turning in. Genetic drift and bottleneck. And I really would like for you to relate this to the kind of problem that you're working with. Genetic drift and bottleneck. Now, if you have a total population, of say 16,034. According to Hardy-Weinberge, in this population, how many would you expect to be heterozygous? So In population genetics, we don't have a parent, we do not have an F1, and we don't have an F2. So that's a controlled cross, actually. But in population genetics, there is a way to determine gene frequency in a randomly mating population of any organism. So this is a potential question that you could see, say, it wouldn't be this complicated, but I just like making things more complicated than it actually is. How would you determine what the expected heterozygote would be? And of course, I didn't give you enough information. And the recessive phenotype, the number of people showing the recessive trait will be, let's say, 305. Of that population, 305 are recessive. So let's work through that problem. Okay? Using Hardy-Weinberge, how many would I expect to be recessive? And essentially, using Hardy-Weinberge means that all these things are true, whether I can prove it or not. But that's the question. If it is in Hardy-Weinberge equilibrium, I can predict the genotype or how many will show, according to Hardy-Weinberge, how many will be heterozygous. And so let's do that. Bear with me. P squared plus 2PQ plus Q squared. In that population, which one of these can I readily identify? In that population, which one of these can I readily identify?
The recessive Q squared.
Q squared, because that's the recessive. Q squared is the same thing as A squared,
and it equals
which is the same as A little a. So that's Q squared. So I can say Q squared equals little a, which equals 305 divided by 1634. That will give me the frequency of Q squared.
So it wouldn't be 16?
Say again.
It would be 610 divided by...
So you're saying the frequency of little a. So we don't know how many are heterozygous because they all look the same. So this is the only one that I can see. So unless I had that other one, I can't do what you're saying. So I got to use a different technique now and predict how many are heterozygous. If I could see the homozygous, the heterozygous, and the other homozygous, then that's what I would do. But I can't here. So I got to use another technique. And so I can't see it. So I can determine the frequency of Q squared.
Which is 0.18.
You say it's, what is it?
0.1866 is what I got.
0.1866, right? Okay. If I took the square root of Q squared, that would give me Q, which is the square root of 0.1866. So if I want to find Q, I would take the square root of Q times Q, right? It would be Q. The square root of Q is Q. What's the square root of 9?
3.
3, because when you do square roots, 3 itself times itself, right? One number times itself. So I can find out what Q is by doing the square root of this, because little a times little a will give me that.
Square root.
So I guess I'm waiting for the answer.
Yeah.
What is it?
0.432.